Good morning, good evening, depending on where you are. My name is Dr. Brian Chung. I'm the Chief Medical and Scientific Officer of the Hong Kong Genome Institute and also the President of the Asia Pacific Society of Human Genetics. So welcome to this very first lecture on the Innovation on the Genomic Medicine series. So let me give you some background about this lecture series. So as all of you know, upon the completion of the Human Genome Project, we know that there are advances in genomics during the last 2 decades, and it has reshaped our understanding towards human pathophysiology, and it's driving a revolutionary shift towards the development of genomic medicine. So in order, in an endeavor to fully utilize genomic technologies and advance the field of genomic medicine in Asia-Pacific region, the Asia Pacific Society of Human Genetics and the Hong Kong Genome Institute actually jointly host a virtual lecture series every quarter. And so we would also invite very prominent scientists, clinicians, and researchers in the field to deliver lectures on their innovative insights and experience in genomic medicine and genomic science. Today is our first lecture, and I'm very happy to introduce our first speaker of the series, Professor Heidi Rein. So let me do a brief introduction. Professor Rein is a board-certified laboratory geneticist. She is the co-director of the program of Medical and Population Genetics at the Broad Institute of MIT and Harvard. She is also the Chief Genomics Officer of Center for Genomic Medicine and the Department of Medicine at Massachusetts General Hospital. Professor Rein is a professor of pathology at Harvard Medical School and the chief laboratory director of Laboratory for Molecular Medicine at Partners Healthcare Center for Personalized Genetic Medicine. She leads the rapid translation of new genetic discoveries into clinical tests and supports several genomic medicine research projects, including the MedSeq and BabySeq projects. Professor Rein is the principal investigator of Clingen, and she co-leads the Board Center for Mendelian Genomics and the Matchmaker Exchange to aid in gene discovery. She is also involved in defining standards for the use of next-generation sequencing in clinical diagnostic and the interpretation of sequence variants through her committee roles as a board member at the American College of Medical Genetics. She is a strong advocate and pioneer of open science and data sharing, working to extend these approaches through her role as chair of the Global Alliance for Genomics and Health. Professor Rein is also a principal investigator of the broad LMM Color All of Us Genome Center, supporting the sequencing and return of results to a cohort of 1 million individuals in the US. And she co-leads GnomAD, the Genome Aggregation Database. She has published over 250 peer-reviewed papers and is the 2022 recipient of the Scientific Achievement Award from the American Society of Human Genetics. Professor Rein, thank you very much for delivering the lecture for us.
Of course, it's my great pleasure to be able to help you kick off this series. It sounds like a wonderful series. I'm going to just start by posting a link to my slides. If anyone would like to download them, you're welcome to. Hopefully you can all see my slides now. We'll get started. And hopefully there'll be some time at the end for questions, and I'll make sure I end in time for that. So thank you. I'm going to talk a bit about how we're really trying to advance genomic medicine through a lot of global collaborations. That's sort of a lot of what I do. There we go. So just to put this into context, I focus a lot on rare disease gene discovery through our Broad Center for Mendelian Genomics, and then also working to build genomic resources to support the community in being able to diagnose patients on a regular basis through clinical genetic testing. So understanding sort of where we are in discovering the causes of rare disease is important as we think about what's left to do. And to date, nearly 5,000 genes have been implicated in at least one rare disease. And that's data from OMIM. But when we do regular testing of patients that we suspect has a genetic cause of rare disease, over 50% of the cases remain unsolved. So we're clearly not, we clearly haven't discovered everything out there. And in fact, there's evidence for more than 5,000 more genes to be implicated in monogenic disease using data both from our GnomAD database, looking at constrained genes, as well as animal models that have rare phenotypes found, but not yet discovered in humans. So many years ago, Daniel MacArthur and I assembled a team at the Broad, which has grown and evolved over the years. This is our current team at the Broad Center for Mendelian Genomics that's co-led by Anna Donaluria, myself, and Michael Talkowski. And we really try to tackle rare disease discovery from many angles: methods, development, recruitment, collaboration, platforms for innovative genomic analysis, and so on. So I'll talk a little bit about our strategies to diagnose patients and discover new causes of disease. And one of the critical things is through academic partnerships. So we partner with many investigators, some of whom are on this slide, working in many different disease areas. And they all recruit their own patient populations, usually in specific disease areas. And then we collaborate with them to help supply the sequencing and analysis and novel methodologies to their cohorts. But then they really take those and functionally validate and publish the papers on the discoveries. We also partner with patient organizations through a variety of ways and have our own recruitment arm called the Rare Genomes Project, where we recruit undiagnosed patients. And these collaborations over the last 10 years have allowed us to really recruit thousands of families from over 50 collaborators from over 57 countries. So it's been a really wonderful partnership and collaboration. More recently, we've been trying to enhance the diversity of our cohort, recognizing that just posting our study on social media doesn't always get everyone out there that needs a diagnosis. And so we've put in some deliberate approaches to help recruit from underrepresented populations. And some of those include how we work with clinicians. So instead of having the clinician give a flyer to the patient and tell them to come find us, instead we have the clinician give us their contact information if they consent to that. And then we reach out and we really help them enroll and engage. And they may just not have the time to figure it out, and we help them. We also have staff who speak multiple languages. We can now support any language in terms of recruitment, and that has been helpful. We also have staff that work nights and weekends, which is sometimes the only time that certain families can enroll. We also now have sample collection services where we can send the services to their home on the nights and weekends or any time during the day to help collect those samples. And these are all ways that we've been able to enrich the diversity of our population and reduce inequities. Another way that we democratize rare disease discovery and diagnosis is making our platform open and free to anyone who wants to use it. Not everyone has a platform to do genomic analysis with a lot of the latest techniques and methods and data types incorporated. And so we built a platform called Seeker. That's our genomic analysis platform. There's a URL. It's now installed on the AnVIL platform, which is a cloud-based platform. And it incorporates all sorts of different genomic data, including RNA-seq, structural variants, all sorts of things. And we're constantly adding features. And this has been used by now over 2,000 researchers from over 100 countries that are performing analysis, because we make this free to anyone who wishes to use it. It's also open source, so groups have also downloaded their own copies of the software into their own environments and been able to support their own workflows. This is that AnVIL platform I mentioned. So we put the current main version of Seeker on AnVIL, where anyone can access it on AnVIL. And AnVIL is a NHGRI, that's the National Human Genome Research Institute in the US, that funds a lot of genomic research. So we put it on their cloud-based platform called AnVIL. And that is a place where both tools and platforms like Seeker and analysis workflows and lots of data all coexist to be able to facilitate analysis. And in fact, putting our data there allowed a collaborator, Aaron Quinlan, to apply a new structural variant detection methodology, was able to detect that a variant in one of our cases that we had shared, and able to make a diagnosis. So it really shows you that working together, can enhance diagnoses and the distribution of innovative tools. So if we look at where we are today, we've been really bringing all of these exome and genome unsolved cases. Most of these have already had clinical testing and been negative. And so this just gives some of the solve rates, both those cases that we deem completely solved and we found the answer. Others, we have a candidate, and so they're still inconclusive until we've been able to prove that candidate. And overall, across both exome and genome cases, we have about a 35% positive rate in our cases. About 45% still test negative, and then about 20% are inconclusive. In the context of those testing rates, there's been over 300 novel gene discoveries, and that's, as I mentioned earlier, working with many of our collaborators who can take these candidate genes to full functional validation. So it's been very successful, but there's still this large proportion that are negative or inconclusive. And I'm going to talk through some of our strategies to address those areas. So in the 45% negative cases, the reason they're negative could be to two main reasons. One, the variant is actually missing from our sequencing data, or two, it's there, but we've overlooked it, not identified it as pathogenic. And those are two big challenges. For the variant being missing, obviously, if you've only done an exome sequencing analysis, then it's an obvious step to move to genome analysis. And one example of a amazingly high yield discovery that simply needed to move to genome was the recent discovery of RNNU42, which is a small nuclear RNA that's probably now one of the most common causes of neurodevelopmental disorders. And that was just discovered, and largely in part because it was none of the exome sequencing assays covered this non-coding nuclear RNA molecule. But there's a very common variant and then a handful of others that are seen repeatedly, and now hundreds of cases have been diagnosed, probably by now thousands. So again, it's really making sure you're looking not just in coding regions, but in nuclear and RNA molecules as well. In total, and we just recently published this data in New England Journal of Medicine, if we took our entire cohort that we've done genome sequencing on and looked at those discoveries or diagnoses that had to be made by genome, and even if we went back to exome, could not make them by exome analysis, we find an added yield of 8.3%. That same exact rate was actually also found in a German cohort that we collaborated with and added to this paper. And the added genome solves are really coming from mainly structural variants, as well as coding variants that have poor coverage in exome because of the capture requirement. And then the deeper intronic variants, those are the three major areas. We also, the detection of tandem repeat expansions is much more challenging in exome data than genome. So that's added a lot. And then I added to this figure because it's just, published the RNA4 discovery, what the added yield would have been for that gene alone, which you can see is incredibly substantive in terms of non-coding RNA variation. So that gives you some sense of what a genome really adds if you look carefully at it. Now, even when we do genome sequencing, the sort of traditional short read sequencing, it still doesn't capture the entire genome. And so we have been applying long read technologies, both ONT and PacBio, to our data sets. And we have made some discoveries, although in most cases, it's been simply the reanalysis of the data that we found things, and then we go back and there it is in genome as well, or even sometimes exome. So to date, we haven't had a huge additional yield from the long read sequencing that really required long read sequencing. I think as we build up better population data sets from long read data, where we can better enable the analysis of this data according in the same ways we use GnomAD to filter out common variation. We just don't have a lot of those resources developed for the regions of the genome that have been intractable to short-read sequencing. So I think over time, as more of this data is generated, these technologies will add yield, but they've been a little slow right now. Probably what's in more important yield is the fact that the variant is there, but it's been overlooked. And RNA-seq in our hands has been actually a really powerful tool. And I'll just give a couple examples. So this is a patient with central nuclear myopathy that had hypotonia, weakness, ptosis, and reflux. The physician actually suspected a particular gene, MTM1, based on a diagnosis of myotubular myopathy, but exome sequencing was negative. So we applied trio sequencing and also RNA-seq on patient muscle tissue. And what we were able to quickly see is that the most distinct outlier was the gene that was suspected by the physician, MTM1. It had very low expression in the muscle tissue. And then looking at altered splicing, we can see that there was skipping of several exons in this proband. And so that was able to point us to the exact region to look. And in fact, there was a retro-transpose on line 1 element insertion in intron 3 of that gene. It was actually mosaic in the mother, which suggests recurrence risk chances as well. But that was able to really direct us quickly to a diagnosis based on the RNA-seq data that could prove the effect of this deep intronic insertion. Now, some of the cases that we do by RNA-seq aren't as straightforward as that one I just showed you. This is a patient with a complex phenotype where in looking at RNA-seq data, there were actually lots of genes underexpressed and others overexpressed, a lot of outliers. And we looked deeply into the intronic regions of all of these underexpressed and overexpressed genes and could not find any obvious candidates. But we did note that the retention of introns in this case was extraordinary compared to all of our other RNA-seq cases. And then if you look, you can see here, this intron is included, but none of the other introns. And so there's clearly something specific going on in certain introns. And it turns out this RNA4 ATAC, small nuclear RNA, actually is part of the U12-dependent minor spliceosome complex, which is involved in splicing a small number, about 0.5% of introns in our coding genes. And so that is exactly why you see these sort of sporadic intron retention. And it's that retention of intron that actually makes it look like these genes are overexpressed because they're giving more signal from the expanded retained intron. And then we assume the underexpressed genes are probably because of rapid degradation of certain transcripts that then making them look underexpressed. But eventually we were able to correlate the phenotype and demonstrate that that mutations in this particular gene were causative in this case. So again, really showing you the power of RNA-seq to help point you to certain etiologies. And overall, we've actually been able to solve 23 out of 170 cases where we added RNA-seq data through muscle biopsies from patients with muscle disease. So that's been a high yield. And then the last way that's probably the highest yield is simply reanalyzing this data. And there's constant novel, new knowledge being generated, both novel gene discoveries, as well as new variants being implicated in disease, as well as new analytical tools like Splice AI that can better detect signals deeper in introns where a cryptic splice site might be generated, or giving you insight to the disruption of an existing splice site. So really going back and looking for new knowledge, looking for new signals is one of the highest yield things we've done. And although we have been reanalyzing our data on a regular basis, it's labor intensive to constantly reanalyze every case you have. We roughly do it about once a year for all of our cases in our research program, but that's in part because we have funding to do these studies. And in a clinical lab, those are not resources that most clinical labs can expend. So we've been really focused on the development of now an automated reinterpretation pipeline. And this is a collaboration with our colleagues in Australia, particularly Daniel MacArthur and Zornitza Stark, our team at the Broad, and then a collaboration with Microsoft. And what we've been able to show is in this top area, the AIP, our automated interpretation pipeline, on both the Australian cohort and our Rare Genomes Project cohort, really recovered most of our positive cases when we were validating it and performed better than some of the existing tools out there. So it displays a pretty good sensitivity and specificity. Also, as we've been now running it on unsolved cases, we're starting to pick up new discoveries. And also, it's important that any tool like this isn't just constantly flagging things that you have to review, because that doesn't save time. And so right now, in the first version, we were getting about 1 candidate variant per 200 cases per month. So that's pretty low in terms of the rate of required review for these variants. And we've now had new solves from this pipeline. But we've also recently implemented a phenotype filtering algorithm to even reduce and increase the specificity even further. So we're going to be implementing this pipeline in a number of settings. One of them is through a project that we've started with the AnVIL clinical resource, where the goal is to really bring unsolved clinical exome and genome cases into a research environment where we can trigger this automated reanalysis pipeline on that clinical lab data. And so we've been working with clinical labs. The first lab we're working with is a US lab, GeneDx, and then working with physicians who are willing to consent their patients they've had clinical testing for. And then they just give us the IDs of the cases, and we bring that data for the physician into AnVIL based on which patients consented. Then we can allow the physicians to access our Seeker platform and be able to look in those cases themselves if they wish to, but also trigger this automated pipeline on that data on a continual basis. And when new things are found, we can notify the clinician to say, take a look at this in your case. So that's a pipeline we're just starting to build, but we think it'll be really important way to capture the clinical genetic testing that's often done once, and then that data just sits there and is not looked at on a regular basis. So those are, some of the approaches that we're doing for the negative cases. Now I'm going to switch to the last bucket, which is we did detect a variant. It's a candidate, but it's insufficient evidence to implicate that variant or that gene in disease. And that could be because it's in a novel gene and we need data to implicate the gene in disease, or it's in a known gene, but it's still a variant of uncertain significance. So this is an area of active work. One of the platforms we launched now about 8 or so years ago was really focused on this concept of gene matching. And the idea is, two researchers or clinicians who've seen patients with rare disease have a candidate gene. Some disruption of that gene that looks suspicious, maybe a de novo variant in a constrained gene, but they've got one case and they don't have enough evidence to implicate it. And I will say, I'm just going to turn to this figure on the right side, a nice figure from the Orphanet group. And what is demonstrated here is that most of the patients with rare disease, roughly 80% of patients with rare disease, have it due to a very small number of more common rare diseases, those in the 1 to 5 per 10,000. But in fact, most rare diseases, over 3,000, are found in fewer than 1 in a million patients or research subjects. And what that means is no individual investigator or clinician will ever see more than one case in their lifetime, probably. And as a result, we really need ways to share data in order to implicate these genes in disease and bring clinicians and investigators together that each have cases that have the same candidate gene. And so we launched Matchmaker Exchange, a platform to do that, about 8 years ago. And it is now a federated network. It was one of the first genomic data sharing federated networks. And what that means when I say federated is no one has to send their data into a centralized location. everybody's data stays in their own database in whatever country or organization it's in, but you allow it to be queried to look for matches by candidate gene. And the only way anyone finds that your candidate gene is in your database is that they also have the same candidate gene, in which case you're matched and you both get CC'd on an email. It says you have matched on this gene, and then the investigators can share more detailed phenotype to decide if it's a valid match. So our Broad Seeker platform is hooked up to this platform. We have about 9 nodes that are linked, as well as some knowledge sources. And over the years, we've submitted our own 1,500 candidates and received over 500,000 electronic match requests that have triggered 13,000 matches. So this system is just operating all the time and many sites. In fact, most groups who don't have their own node hooked up to Matchmaker Exchange can just go in and use GeneMatcher, register an account, and submit their candidate there. And that's where the widest use is today. In fact, there's over 15,000 candidate genes in this system from over 200,000 individuals from 13,000 contributors from 103 countries. So I would argue it's probably the widest used tool in our rare disease discovery arsenal of tools. And it's really yielded an incredible number of gene discoveries over the years. And that whole platform was built using standards developed by the Global Alliance for Genomics and Health, which is a really important standards organization. However, sometimes you put your gene candidate into Matchmaker Exchange and don't get a hit. And then what do? There's not much to do in terms of that space. So we're really working on the next phase of this, so that start that I just described is really what we call two-sided matching. So you're brought together when both groups have a candidate and you're matched. But I just told you, well, what if you have a candidate, but no one else matches with you? Well, wouldn't you like to query all the raw genomic data in the world and see if anyone else has either that specific variant and get their phenotype back? Or maybe you want to query and say, does anyone have a genomic case that has a loss of function variant in this gene that I just discovered as a candidate? So we want to really move to this query approach to access raw genomic data and the variants in them, even if no one has identified a candidate. And somebody in the future will have zero-sided matching, and we can query all this genomic data for hypotheses we don't even know today, but really we're focused on step B right now. And an example of this, the same group that brought you GeneMatcher, led by Nara Sabrera, they've launched Variant Matcher, where you can just log into their system and enter a variant. And if it's found, like in this case, I entered this variant, it will give you a list of the patients and their phenotypes. And what I was able to quickly do for my case with the phenotype shown at the top is quickly rule this out as causal, because the 4 patients with phenotypes, none of them were a match. And then there were 4 patients who were actually unaffected parents that were clearly not a match. So it's equally important to rule out candidates as it is to rule them in, because if you rule out, then you can move on to other ideas. So we're now starting to build the next federated network of genomic databases to allow this variant querying to happen at scale. And in fact, the first node-to-node linkage has happened between Variant Matcher and the commercial Franklin platform by Genomics. But these are all groups listed here that are working. We have a monthly meeting every Thursday morning to work to bring new nodes onto this network. It's being powered by Global Alliance Standards, the Beacon version 2 API that was built by the discovery workstream of the Global Alliance. And this is a collaboration with the Global Alliance to really help support and launch this platform, and using some of the other standards in terms of security and access that we can build into this system. So we're really excited to get this platform launched. As I said, the first 2 nodes are now live. So if you go into Variant Matcher to query, automatically it will default to also querying Franklin at the same time and returning if there's that variant in Franklin without you having to search 2 separate databases. Likewise, if you're in Franklin and you use that platform, automatically it's querying Variant Matcher, and this is just 2 different examples of 2 different variants where it says variant doesn't appear on Variant Matcher, or here, variant was submitted 726 times, 144 were homozygotes, on Variant Matcher, and then this hyperlink directly sends you over to Variant Matcher where you log in and get the phenotypes. That way you only have to search if the data looks interesting. Now, if I was doing this particular variant, I'd say, oh, that's too common to be a cause of rare disease, and quickly rule it out and move on. But if it was like, oh, seen once or twice in Variant Matcher, then I might log in and look at that data. It might help me. And in fact, since this was launched in 2024, there have been, so Variant Matcher is a smaller database with a smaller set of users, 362 queries of Franklin, and the reverse 1.3 million queries from Franklin back to Variant Matcher. So you can see the power that these connections will have over time as we expand this network. I do want to just mention another element of our GnomAD database. So I co-direct the GnomAD platform where we share aggregate genomic data. And in the fall, we released GnomAD version 4, which added a whole lot more data. So now we're up to 800,000 genomic data sets being shared in terms of aggregate allele frequency. But I do want to point out that a lot of the data that got added in this last tranche was actually a fairly large set of European ancestry individuals because we added the UK Biobank data set to GnomAD. And so you can see the European data went from this about half the data set to a large fraction. We did, however, have an incremental increase in non-European data as well. But what I want to focus you on is not the sample growth, but looking over here at the number of variants that had an allele frequency over 0.1%. And that's high enough to exclude as a cause of rare disease for the most part. But what you see is the number of variants that we could exclude from European data sets almost was unchanged. Yet even that small incremental increase in non-European data yielded a much higher number of variants we could filter out and exclude from rare disease causality. So what that's saying is we have largely saturated our understanding of common variation in European individuals. We have not come close to saturating that knowledge in non-European populations. And for that reason, it's really critical that we gather more diverse data sets from other non-European populations, because it will dramatically reduce the number of variants we all have to look at when we're trying to solve rare disease cases. So we are very focused right now on bringing non-European data into GnomAD, and the next launch, V5, will include the All of Us data, which has been intentionally recruited for non-European individuals. Over half of that data is non-European. And then we have launched a new project called Federated GnomAD, and these are some of the countries we're currently working with, where the goal is to teach those countries how to process their data like we process the GnomAD data, because they are unable to physically send that data to us at the Broad where we could process it ourselves. And for data sharing regulations in these countries, they can't send it to us, but we're going to work with them to teach them how to QC all the sample and variant data to be equivalent to the GnomAD processing pipeline. And then they can share the aggregate allele frequencies that we can merge with GnomAD and give a much more diverse data set to the community. So that project also has a collaboration with the Global Alliance. We're working on these federated approaches. So that project is underway, and we hope it will yield fruit soon. But that really brings me to talk a little bit more in depth about the Global Alliance. It's a critical data sharing organization. However, I do see that there was a question in the chat. Variant matches is very cool, yes. Is it possible to match with transcript isoform, say skipping a canonical exon, because the same transcript may be probably derived from the same essential splice site, but just site a bit shift. So the truth is, typically you're querying a genomic coordinate. So if there is a variant, regardless of how it's causing an impact, whether it could be to one or two or more transcripts or distinct transcripts, you will find the genomic variant in a data set. And then that will share with you the existence of the variant and the phenotype of that individual. If it is a match, that it builds some evidence for you, and now you can continue to explore the functional impact of that variant, which may be disruption of an alternative transcript. So the genomic data is really finding the added cases and their phenotypes, but then functionally figuring out how that variant leads to disease would be data you would generate from things like RNA-seq. And now you have more samples to get more tissues and more experiments on. So that's how we sort of see it. First, like find all the cases with the variant using genomic coordinates, then bring those cases, those phenotypes, those samples into your studies to prove the validity of that finding. But that's a good question. So I just wanted to, mention Global Alliance is really a standards building organization where we bring the community together. We have driver projects in many areas: rare disease, complex traits, basic biology, cancer, or some projects covering all of the above. And those driver projects work with our work streams to develop the standards needed to share data and advance genomics. And we take that input and then we output the standards that are tested by our driver projects and other members of the community. And that's really been a critical pipeline to enable us all to use data globally. Because if your data is formatted in distinct ways or annotated in ways no one else does, it's really hard to share that data productively. And we also, in addition to forming this whole system for standards building, we also have a national initiatives forum as part of Global Alliance where we bring those countries together that are doing national genome projects. And I understand that's happening in Hong Kong. So these groups can come together and share best practices. In fact, we're building a national initiatives toolkit with all sorts of different standards and policies that help with government strategy, engagement of patients, clinical tools to gather phenotypic data and other information, how to consent and return results to participants enrolling in biobanks, providing the technical and data analytical tools, and also participating in genomic knowledge curation across countries. So these are all things with standards, some of which have already been developed and others we are working on to really empower many countries launching national programs. And I've also just recently launched a clinical genomics laboratory community. So we have about 5 different communities of focus based on common areas of interest. We have them in infectious disease, cancer, rare disease, and neuroscience. And now the 5th is this clinical genomics laboratory. So if you're part of a clinical genomics laboratory, you want to participate in this, you can sign up through the GA4GH website. So that's a bit about how we tackle the VUS area of this through platforms like Matchmaker Exchange, this evolving variant level matching federated network I mentioned, through building out GnomAD with diverse data sets. Of course, functional studies are critical in that end validation, which I won't talk too much about today. They tend to be unique and specific to the gene and disease area. But with a constant evolution of evidence for genes and variants, how do we keep track and ensure the community research are accurate for widespread use? So many of you know I'm involved in the Clinical Genome Resource, or ClinGen program. We have expert panels that have formed in nearly every disease area and engage really volunteers across the entire globe in terms of participating in expert curation. And that's a critical effort. For those of you who aren't already involved, you can get involved either through becoming a trained biocurator and being assigned to any group if you're earlier on in your career and don't have an area of clinical expertise, or if you do have a specific area of expertise and want to join a specific expert panel, you can reach out to the coordinators of that group and ask to join them. So a lot of people in the community join and participate. It's a wonderful way to engage and contribute to expert curation. We also, in the last couple of years, launched a new resource called GenCC, and this is really trying to get community sharing around gene disease validity curation. So is this gene that's been published as a cause of X disease, is there valid evidence for that? And so we really brought together many different gene level resources, some public, others private databases in clinical labs, and brought them all together on one platform. And this is just a quick peek at the GenCC database. I like to call it the ClinVar for genes, but you can look up any gene you want and see what diseases it's been implicated in and with what level of evidence by what group. And then when there's discrepancies in those scores, we work within the GenCC monthly meetings to resolve those, and we've been resolving many of these discrepancies, as shown here. So that's one resource. If you're not familiar with GenCC, I encourage you to use it and even encourage you to submit your own. And we're actually working with the Gregor Consortium, the NIH-funded rare disease discovery program, to get the groups to actually submit their early gene discoveries even before they've been published into GenCC, so that data gets out to the community even quicker. Of course, ClinVar, we've been working with the ClinVar group at NCBI for many years to create an environment that is really conducive to variant level classification sharing. It now has over 4 million submissions from groups all around the world. However, one of the challenges we deal with all the time is that, and I've alluded to this in the rare disease pipeline we have, is the rate of VUSs. And in fact, our own cancer program has tons of VUSs. They're constantly being reclassified. We have insurance companies that are refusing to pay for genomic testing because they're fearing too many VUSs getting returned to patients. It is the highest category of classification in ClinVar, so it's really creating a challenge for us. We did a recent study and we're able to show that the highest rate of VUSs is not generated from exome and genome sequencing, but in fact from multi-gene panel testing. And you can see here, as the size of the panel goes up, the rate of VUSs in these results goes higher. And no surprise, you have more genes, more content, more VUSs to be found. But interestingly, despite being higher content in exome and genome, that VUS rate goes dramatically down. So it's counterintuitive, right? Why would that be? But it turns out it's just around the policies of reporting variation in these tests. In multi-gene panels, it's standard practice to not correlate with phenotype. In fact, often we don't get phenotypes for gene panels. And you classify every variant you find, and you report VUSs and above. And that's the standard practice. In contrast, in exome and genome sequencing, we require a phenotype. You can't interpret the data without a phenotype. And then you only report those VUS where you're very suspicious that they might be a candidate cause, because they correlate with phenotype and they look more suspicious in terms of the evidence for pathogenicity. That is why the VUS rate goes down. And in fact, for two of the groups that use subclasses of VUS, what you see is this low evidence category of VUS low, where a quarter of the variants reported in multi-gene panels never reported for exome and genome sequencing. So it really emphasizes that this VUS subclassing would really help us. And in fact, in the new ACMG, AMP-CAP, Clingen guidelines that we're working on right now, we will actually use a points-based system, and that will allow an automatic subclassifying of VUSs into a low, mid, and high bucket. And those are the terms that we're going to use in the next release. And in fact, 4 labs that we've been all working and sharing our data, because we've been using subclasses over the last decade. And what we've been able to show is that variants in this VUS low category have never moved to likely pathogenic or pathogenic in the 15 years we've been tracking it. And in fact, even the VUS mids largely move down to pathogenic, and only a small fraction move up. However, the VUS high category, nearly half of those variants moved up to path or likely path. What this shows you is that it's incredibly valuable to use these subclasses to help guide follow-up that might occur after you do a genetic test, or to potentially even exclude the VUS low from the patient reports because they're not yielding additional classifications in the path likely path category. So we're actually writing a second guideline with another work group that I'm co-chairing on when to report VUSs and how to use these new subclasses that will help guide the community in what to do once the sequence variant classification version 4 comes out with these new tiers. Will help guide how to use those tiers in reporting. And with that, I'm going to just take the last few minutes to touch on a few things we're doing in genomic medicine implementation. I lead a small but mighty team at MGH called the Genomic Medicine Unit, And we really work to engage our clinical stakeholders, identify pain points in delivering genomic medicine, and deliver solutions. We also work with representatives of every clinical department and division across the hospital, both at MGH, where I'm primarily stationed, but we're also merging with Brigham Women's Hospital, and we have representatives of all of those departments at the Brigham. And we really work together to solve challenges. And some of the biggest challenges are the high volume of patients with cancer risk, meeting professional guidelines for cancer risk, but not having enough resources to get these patients tested. Also, preconception carrier screening. So we launched a preventive genomics clinic to serve this high patient need in our cohort. And it was doing well, but we just didn't have enough physicians to see all the patients. So we actually discontinued this clinic last year and moved on to other strategies to support the high volume needs of genomics in our patient population. We launched an e-consult service so that clinicians could ask quick questions of our experts, and we could help guide them. And then often those answers helped lead them to using that information in future cases. And we published a paper recently showing the outcome of that e-consult, and we're hoping to expand the service in the future. The other thing we did when we ended the preventive genomics clinic is we switched it to a preventive genetic counseling service. And this is work led by Leland Hall, who's a primary care physician. And basically now our genetic counselors are working directly with primary care physicians to help in the education, consent, and return results directly during the primary care visits. And you can see that this is also addressing these high volume needs in cancer and preconception carrier screening, as well as the direct to consumer test reports that come back that patients bring to their physicians. But we're rapidly expanding this service because it's really found to be a high yield way to allow lots of patients to benefit without requiring a referral to a genetics or other clinic, which often is where you lose particularly underrepresented populations. So with that, I'm going to stop there. We have about 12 minutes for questions. I hope you'll ask many tough questions of me. I do want to thank many people in my different teams that work on many of the projects that I talked about, as well as our funding sources. And again, here's the link to a copy of my slides if you want any of the links that I put in before. So with that, I'm going to stop sharing these slides and happy to answer any questions.
Thank you very much, Professor Rin. This is really a great treat for all of us in Hong Kong and in the Asia-Pacific region. Your talk has covered area from primary care to tertiary care, from gene discovery to clinical implementation, from work in Harvard and Broad Institute to the whole United States, and then your global initiatives in joining hands together to work for patients with genetic disorders. This is really a great presentation. I think I would take the privilege of asking the first question. As you know, that we are now implementing the Hong Kong Genome Project. And in this project, we are using whole genome sequencing to find answers for our patients. And I face this big headache of a timely return of results and timely analysis for our patient. And I see a lot of good efforts, including automations and so on. Can you share more about the rationale, how you actually speed up the return of results to patients?
Yes. So one of the things that we did was make sure when we're taking samples in, we actually have those samples sent to a clinical lab first, who extracts the DNA and then sends an aliquot to us for our research projects. But what that means is if we identify a cause of disease, we want to return results, we don't have to go collect a new sample under clinical regulatory procedures because that sample's already been collected in a CLIA lab and stored there. And we can simply go back to that CLIA lab. We partnered with a particular lab. It actually was the lab I used to direct at Mass General Brigham. They do, Sanger confirmation. They also do digital droplet PCR to confirm structural variants if we find those as causal. And so our team assembles the evidence for the variant and the gene. If it's a novel gene discovery, we draft the text of the report, and then we partner with our clinical lab that's doing the confirmations. And actually board certified geneticist in my team, Chrissy Austin C, is the one who signs out most of these. She works both in our research program, but she's also a board certified clinical lab director. So she signs out the cases in our clinical lab so that we really make that a much more streamlined process, ensure the evidence that we're collecting on the research side is part of the clinical report, and it can help get those out. We also, we partner, so when those patients are going to get a result back, we contact them, we say we found something that we think might explain your disease. Do you have a physician that we could work with to return those results? And we use their own physician to return those results to them. And that physician actually officially places the order with the clinical lab we work with, but we pay for it. And so that's a free return of result to that family. And then if they don't have a physician, we have another service that's a sort of a service that can confirm results in any state in the U.S. And we pay for that service to be the genetic counselor and return those results and the ordering physician. So we kind of set it up for every scenario we can to make sure those patients and families can get a result back with the guidance of a physician and a genetic counselor to help with that return. And sometimes we do return variants of uncertain significance, but things that we think are more likely to be causal, because sometimes those families then are part of pushing the field forward and finding other families and engaging in that building of evidence process. And so we will return clear positives, but we also will return those candidate variants or genes and allow those families to participate in the evidence generation and then update over time if new knowledge is generated.
Thank you very much. It seems that you have built a very effective clinical research partnership in order to do things efficiently. I have scanned through the Zoom list. I do not see any question yet, but I know that Dr. Ma Wei, our lead scientist of the Hong Kong Genome Institute, have gathered some questions from the scientists and the genome curators working in the Hong Kong Genome Institute. Dr. Ma, would you like to ask some questions?
Yes. Thank you, Professor Rim, for the wonderful talk. We learned a lot of the new developments from you. So it's very helpful. And actually, my team is actively asking questions. I will represent them to, just to make things more clear. So the first question I like to ask is, actually for the upgradable VUS analysis, we also perform a similar exercise, summary calculation in our project. So we also see a very similar trend. The so-called VUS mid is never upgradable. It's always those hot VUS, what we call the hot VUS and high VUS, that we can upgrade them to lightly pathogenic. Mm-hmm. So my first question will be, for your Reanalysis pipeline, is that's mainly used for the upgrade, I guess. So is that like can we try it in your Seeger platform? And in that reanalysis process, will you separately handle those genes that has a newly published gene-specific guidelines, or you just mix everything up in your reanalysis the pipeline and then perform the annual reanalysis in a big batch of samples?
Yeah. So right now, we are working on Dockerizing that pipeline so it's more distributable to many groups and will be easily accessible. So it's been in a development phase working with our colleagues and Daniel MacArthur's team in Australia, as well as our Microsoft team. So we're working on being able to distribute that pipeline so that others can use it, and we've been adding additional features to it. But it doesn't, you don't set it necessarily to reanalyze for a specific candidate variant or gene. It's really looking across the entire genomic data set for that case to flag any new things. Usually the things that we've already flagged as a, VUS hot or, suspicious VUS, we're regularly checking those for those cases on an ongoing basis. And have sort of other strategies to routinely, you can follow a variant in ClinVar, for example, and anytime any new submissions come to that variant. And this is one reason to submit your variants to ClinVar, is once your variant is in ClinVar, then you can track it and monitor it and have it tell you if anyone else submits on that variant. So it's an added reason to always submit all your variants. And we submit everything to ClinVar, even when they're candidates, VUSs, and we just put what evidence we have. Because sometimes we get matches not through matchmaker exchange, but in fact, other people seeing our variants in ClinVar, and then they reach out to us and it turns out they have a similar case. So these are many different ways that you can share your data, both to be monitoring new evidence over time, as well as solicit others to reach out to you if they have similar cases.
Yeah.
So before Dr. Ma asks his other questions, I see one question from the chat room. Professor Sutana Faraz from Indonesia would like to ask a bit about the genetic counseling in primary care clinic. Professor Rin, you mentioned about the services to patients with cancer, preconception carrier, and also direct-to-consumer testing. Professor Sutana, who is an expert in genetic counseling in the region, would like to ask you about what about services for patients with rare diseases and other types of cancer, for example, colorectal cancers. Do you offer services through your primary care network?
Yes. So So that whole implementation team where we have representatives from every division and department, so we actually have an entire network of departments that are offering genetic testing in their own disciplines. And so all of the rare disease patients are going to the relevant clinic. Now, if they're syndromic and aren't just an isolated cardiology or kidney or, whatever disease, then they go to our medical genetics clinic that sees any patient with a suspected genetic disorder. So that actually is a fairly robust pipeline that's been servicing many patients with rare disease. Over the last few years, there have been a few clinics that haven't been as robust in their use of genetics, and we work closely with them to, some of them need like part of a genetic counselor, like 20% effort, but they can't afford to hire a whole person. So my team hires someone and then distributes 20% to you and 5% to you and 10% to you. And this way we help some of these clinics that aren't as familiar with rare disease genetic testing in their discipline to actually launch genetic testing. And we've helped our nephrology clinic do that, our pulmonary clinic do that, our allergy and immunology clinic do that, because we distribute a genetic counselor to support multiple clinics and allow those services to be accessed. We also have a dedicated hereditary cancer clinic. Actually has 14 genetic counselors. It's a very large clinic. It services both colorectal, breast, and any other cancer. So the patients who actually have cancer are going to the specialty clinic, the cancer clinic. What I was talking about is a set of patients that have been lost in the system and have nowhere to go because they don't actually have cancer, so they aren't being prioritized to be seen in our cancer clinic, and they don't have an actual rare disease to then go to one of these other specialty clinics. And the primary care, clinics don't know genetics well enough to actually offer these preventive genomic testing. So that's why I was focused on that. It was sort of a gap in our system, but it wasn't meant to say we don't have rare disease testing happening all over the place in our specialty clinic.
Thank you very much. Because of the time, maybe one last question from the chat box. Dr. Johnson Yee, I would like to ask, is AIP development based on current ACMG interpretation guideline V3, or you are already considering some important new concepts being implemented in the V4 version?
Right. So just one thing to clarify, that automated pipeline is not a pipeline to automate the classification of a variant. So it doesn't actually use either of these standards. It's really trying to identify novel causes of disease in a patient's genome or exome. And so it's actually querying databases like ClinVar and GenCC, as well as applying annotation algorithms like Splice AI and looking for very high scores, or other annotation sources that might bring a candidate variant out of the background of noise of variants, and then tee up those candidates. So, as new genes get discovered and deposited in OMIM and GenCC, new variants classified as pathogenic and likely pathogenic in ClinVar, or new tools and algorithms being applied, that's what it's scanning for, is the presence of these candidate variants. Then once we get notified, oh, there's been a hit, your case has something interesting, then we would look at that variant. We would apply the current ACMG guidelines and variant classification to that variant once it's been teed up. So it's really a primary scanning or screening algorithm to find new things, so that we don't have to one by one reanalyze every case every year ourselves, and they're really flagging things that we should pay attention to.
Thank you very much for the clarification. I think all of us, ladies and gentlemen, would agree that we had a very great 60 minutes treat from Professor Reed. Professor Reed, I hope that this is just the beginning of a new friendship between your team, your group, and Hong Kong, as well as Asia Pacific region. We are going to definitely invite you more frequent to our activities, including lectures such as this one, especially after the launching of the ACMG Re4 classification guidelines. Hope that you don't mind.
Yes, of course.
I would like to thank you once again for giving us this lecture. And friends from Hong Kong and Asia Pacific region, have a great evening. Thank you very much for attending this lecture. Thanks so much. Goodbye.
Thank you. Take care.
Thanks a lot.